Yeah, just so yeah.
How about that? All right, there we go. Okay. This is a meeting to discuss the bottlenecks of aging, the second one. It's gonna be slightly different. There's two ideas that are gonna be discussed here. One is going to be related to heat shock, and the other one is going to be related to the fundamental nature of aging. The first one is a very practical idea that is low to the ground. I think it's a good idea for something Vita DAO could do. The second one is also something that Vita DAO might be interested in, but so far, very few people have really understood the idea that, so I'm not sure how it's going to be received. So we're going to start with heat shock. And to just give an overview, heat shock is a process where your body, through either getting overheated or through specific other chemical stresses like, I don't know, acid, like spicy foods and stuff like that. Submit Keithan. There's a reaction, and the reaction is that a bunch of heat shock proteins are released in your cells that stabilize protein configurations throughout the body. This is a well-known phenomenon, and often it's studied in the context of either heat stroke, heat in a sauna, or like hot tub and stuff like that. But turns out that you can also probably, it's very likely that you can induce heat shock using just a hot shower. The issue is that nobody knows what temperature or how long to shower for in order to induce a heat shock reaction. So my idea was basically what you do is you build a shower head with a thermometer on it. And then you run a series of experiments on a person or a group of people, whereupon you measure the heat shock protein production after a hot shower of varying temperatures and varying lengths. And then it's basically, you just say, hey, here's what happens. If you want to induce a heat shock reaction of this level, take a shower for this long. And it's a very simple experiment. It's very straightforward. It's all very well-known science. It's just that nobody's done it. And I think that what you do then is that you would sell those types of shower heads. There are, I'm sure, out there shower heads that already tell you the temperature, but you're selling two things. One is the information, and then about how long and how hot to take the shower. And number two is that there's going to be people out there that are going to say like, well, if this is the shower head they use in the experiment, then I'm going to go with that one, right? Most people right now don't have any clue how hot their showers are when they take a shower. They don't really know. And because most shower heads don't have thermometers on them. So if you make one that does, and people are going to have a choice to replace their shower head because they want to engage in this easy activity, then they might as well go with your shower head that you used for the actual experiment rather than a shower head that somebody else has produced that might be calibrated slightly differently or whatever. So that's an overview of the idea. I'd like to hear your thoughts about that before moving on to the next thing. Yeah, Tim.
So this kind of already exists, this 8-sleep bed, right? This thing has temperature regulation. So maybe one way, instead of like trying to find somebody who could do a shower head for us, like their premise is essentially that, temperature controlled mattress, which is what this 8-sleep company does, that's actually like better for your health, right? So I don't see any science that backs up that claim. A lot of Silicon Valley types, there's a lot of interest in this 8-sleep mattress. And I actually even considered buying one myself, but there's no science that actually validates it. Right. So I think that would be a good way, so we don't have to kind of come up with the whole field ourselves. Of course, I'm not sure they'd want to know what the result of that science is, but it's just weird because like they had essentially kind of what you just described, is not without water, but with heat, like the heating device is actually basically right next to your head. And basically part of the reason why I didn't go through and buy one is like, well, I don't know what the long-term consequences are of like having basically a heat source just basically irradiating your head all night long. So, I mean, everyone thinks it's a great idea, but yeah, I want to know the science behind it. So my suggestion would be to partner with them so that they already, yeah. But I think how you measure success, I think, is always the challenge with these things. But yeah.
So for this one, all you're trying to do, you're not really necessarily trying to measure lifespan extension with this concept. You're only trying to measure the in trying to induce a heat shock reaction. This is a very straightforward measurement. You're measuring certain proteins, like soluble heat shock proteins that get produced. So it's blood samples. Yep.
Yeah, so I would suggest that so I think in this bed, they also have like heart rate variability. So doing blood tests, right, is not going to be there's not going to be a low feasibility for that. So heart rate variability is a measurement that is feasible, right? I think this bed measures it. It's on all these smart watches and stuff like that. Essentially, it measures the distance between your RR intervals and your on an EKG, essentially. And so that measure, your variability, heart rate variability, basically goes down with age. So one way where we don't have to involve any, blood tests or external things or things that we would, we have to keep the feasibility there, right? So I think one way we could do that would be to just literally do people before and after. Maybe the study's already been, maybe this 8 Sleep's already done this because, essentially people are, well, no, I guess they would have to be able to measure the heart rate. They'd have to basically measure before and after buying the bed. So, but what I don't want to get into is like a scenario where we have like right, where we'd have to find the shower head, find the blood test, no blood tests is, something where the cost would be low. I think we got to think in terms of things that are, of high feasibility to get a lot of participants.
I feel like doing blood tests, it's not expensive and you're not going to need a ton of them,
it's just like somebody has to go to a, facility, right? It has very poor feasibility compared to things that can be measured on smart watches or like Keith is saying, visual signal or something which doesn't. My bias is honestly, even though I'm a molecular biologist, and so I would love blood tests and all the genes and molecules and stuff. My bias is that the revolution's gonna it's all gonna be computer generated. It's all gonna be digital, and because that's going to be much more feasible, right? Computer stuff is way more feasible. So
if I can add some context here, Tim.
Keith agrees with me on this.
Yeah, I was just going to say, because I was involved a lot with a lot of the testing that was going on for COVID-19, the machine learning assisted testing, and things like heart rate variability were like immediately tossed out because they were just confounded by too many things. But what was the promising avenues were things like face and voice, right? So if there was, I actually haven't thought about it before, Aaron, but like if there was some sort of visually distinct signal, a certain kind of flushing that lasts for so long or whatever, that is like a visual correlation to heat shock having occurred generally, that could be a promising, because I could see a shower head that might have some sort of like, assistant, camera somewhere. Of course, you'd have to, figure out the privacy concerns with that, right? But like, you hold your wrist up to next to the shower head on the side or something right after you take the shower. Something like that could work here, where blood tests and stuff, I think, to Tim's point, that takes it out of the realm of like feasibility.
All you need is a shower head with like even an alcohol thermometer just sticking out of it. That you just need to know how hot your water is, and then you need to know how long to stand in there. And if you can compare it to a database, like a table of, here's how long versus the heat shock reaction that's been generated. And ideally, you would do this for several different body types because people are going to have different, varying amounts of fat is going to determine how much your heat shock reaction is going to occur. And I think, I don't know, if you think that a blood test is untenable, I feel like that's the only way to actually do something real. Everything else, if you're not measuring directly those heat shock proteins, I don't think you're going to get a good proxy for.
Oh, for testing, probably, sure. I'm saying like if it comes time to like sell a product, what would put me over the edge is if I knew there was something I could see while I was in the shower, that then, something goes off and says, hey, you've done it. You've entered heat shock.
Yeah, it's difficult. I mean, if there was but that'd be like basic research level, is to figure out a test that will let you see heat shock progressing.
Well, I'm saying that's
real
where I
time.
think the secret sauce is to come up with a corollary between an easy physiological signal and a more invasive one that you did in the study. So you say, hey, there's a 95% correlation between the blood test being successful and this visual signal happening. Then when you sell the consumers, you could say, hey, if this happens,
I mean, you could try to do based on like feel or something, or the amount of, like, your skin will get red, inflammation, but that doesn't work for everyone's skin. So I don't know that. Yeah, that's a question for sure.
yeah, I'm not saying it's easy. I'm just saying, like, I think that would matter on the consumer side of it. To your point, maybe that's the second level after the basic sciences there. Just pointing that out.
Okay.
Yeah, I mean, my points are about like I said, feasibility and having, and also the heat shock, right? It's not, heat shock is good or bad, right? It's not like always good. It's not always bad, right? So what you really, I mean, you do kind of want, whether your body's in a better state or a worse state. And I think if you can measure things in the blood, you just don't know at that moment whether you're actually in a good state or a bad state. So, and that's where the things like the heart rate variability come along. Though, yeah, I agree with Keith. It's, it is integrating a lot of signals, but long term, I mean, if your heart rate variability went down over the span of months, it's not going to be something like on the span of days that, I think you could reliably trust, but on the order of months or years, of course, that's a pretty robust signal that responds to your health. So, yeah, I think in the moment, signals that come up every day or whatever, I mean, those are pretty unreliable signals. Most things that really matter don't change that much. So, so I think that's the challenge with any molecular test. So.
Okay.
So I think
so if this kind of project is not
I think your general point is basically, is heat good for you or bad for you, right? Or
no, you just want to figure out if you're trying to induce a heat shock reaction at a in an intermittent
but so ultimately, is your thing is this a therapeutic thing, or is this like is this bad?
You're trying to get basic regeneration. You're trying to improve your daily basic regeneration through inducing periodic heat shock reactions. So the thing about hot showers is you have no idea whether you're actually passing the threshold. You don't know to what extent the heat shock reaction is occurring. Everyone's, they say saunas and whatnot, but and exercise will also do it, but in the hot sun. But you don't know to what extent. Nobody knows like how it's but if you get blood samples and you test it the same way that scientists are currently testing for heat shock, then you'll know.
Yeah.
So
I mean, again, I think this will be an interesting area to partner with 8 Sleep on, because I think their thing is a constant heat over the 8 hours that you're sleeping or whatever, but you can immediately imagine, programming their beds to be something that would do, essentially intermittent, like you were describing and I just think again, just to feasibility, working with somebody we already know who's kind of has a vested interest in, essentially what you're saying, intermittent heating is a therapeutic modality, rather than us trying to come up with a whole, right, we're essentially the money people, right? So we're not, I think our ability to really design studies and, orchestrate studies we're going to be somewhat limited in that. I think we'd probably better be just go with people who have vested interest in seeing the results. If there is a shower company that has a smart shower, we could do that too. But yeah, I think this 8 Sleep thing seems maybe something to consider.
Okay. Avi, you have your hand raised?
Yeah, I was just going to quickly ask, Aaron, you probably have done this, but controlling for socioeconomics and things like that, there are parts of the world that are always in heat shock. I'm in Oxford today, and my indoor temperature right now, I'm looking at it, it's 31 degree fucking Celsius, which I realize if you're from the South in the United States, where I used to live in Texas and Oklahoma, 86 degrees, 90 degrees Fahrenheit is not really that big a deal for you guys, but you have air conditioning. I have a fan blowing on me and it feels like a hairdryer. Not in the cool setting, by the way. So there are parts of the world closer to the equator that are always in heat shock. They don't need the shower and things like that. And of course, I do mention controlling for socioeconomics. So there are parts of India that are much wealthier than parts of Oklahoma that I have lived in. Certainly Mississippi and other parts of the United States. So do we have any epidemiological data? I know, like, the only thing that I know of is looking at type 2 diabetes rate with temperature. It leaves out Middle East, but it looks at US distribution, population distribution, and temperature distribution. And it was a study that was published, I think 6 or 7 years, more than that ago in NGEM, New England Journal of Medicine. But besides that, I'm not really aware of lifespan data controlling for socioeconomic factors and other factors. I don't know, Aaron, if you have looked into this.
So we don't, for this project, is like that goes into deeper just heat shock theory in general. So there's a disparity between, is it good to be in heat shock all the time versus to have it, staggered, right? You induce small amounts of heat shock, like it's like intermittent fasting basically, versus being in starvation. Starvation, we all know, is pretty bad, but intermittent fasting seems to be pretty good. So to what extent is a constant slightly hot temperature inducing heat shock? I don't know, and I don't think anybody really knows, because I don't think anybody's looked at the heat shock protein, soluble heat shock proteins in the blood of just people they're just sort of like, okay, these people are in a cold area, these people are in a hot area. What's their baseline difference, right? So I don't think anybody's looked at that. That's a lot bigger project, because you're looking at like measuring probably thousands of people, and in order to get actual data, you do blood samples, right? But that's obviously, that's a really good idea in general. Like somebody should do that. I don't know if it should be this group, but somebody, like, that's a really good idea. And then just like figuring out how the deeper sort of truths. But I figured that if you do this thing first, you get a shower thing going. That's a very simple, easy concept that everyone could participate in without much effort, because you're just putting a thermometer on a shower head. It's like the most simple, modification. And then with a tiny bit of information about how long and then how hot should it be, you can do something tangible, and if you have data that's trustworthy, then you can participate. Like anyone with a hot shower can participate. It's not just for super rich people. It's everyone can participate. It's a very low to the ground thing, and then you start that thing rolling, and then if it actually takes off, or if it goes somewhere, now you get deeper into it. Now you can start doing these other studies of like, okay, well, if you're doing this thing in a hot climate versus a cold climate, are your benefits going to be different? How often maybe should you shower in order to induce heat shock reactions? Is spending too much time in it a bad thing? Possibly. We don't know. Nobody knows these things. Nobody's asked these questions. So there's a lot of follow-up that could be done. The real question is this something that VitaDAO seems like it might be interested in? If I really don't think you need to partner with anybody in order to do this. It's the most simple invention, but partnering with somebody who would just make the thing seems like might be a good idea, in the sense that you're just outsourcing your manufacturing to somebody else. But to partner with people who already have a vested interest in it, you're basically saying like, I'm going to split the IP again with people who really don't need to be involved, because this idea is just dead simple. Like you really shouldn't you need a lab that will they'll process samples for you. They don't need to have IP rights. They just need to process samples. They're like a vendor, right? And you need a simple manufacturer to make these shower heads. That's another vendor. You don't need to share the IP with anybody. You just need to, you can have full ownership of it. It's just that, I don't know. It's just the question is, do you guys is this something that you're interested in? So, I don't know if you get, before we move on to the next topic, I just wanted to like sort of figure out if this is something that's within the realm of possibility, Tim.
Yeah, I mean, I think it's, I think it's definitely all possible. I mean, I think, again, the idea that heat is, has therapeutic properties is of course important. So I think it's just, what does the execution look like, and, who's going to do it? And yeah, my bias would be to get somebody that has a vested interest in doing it, where we would, maybe provide a little bit of funding, but that essentially they do all the study, and we get, some of the reward for helping facilitate it. But I think this could take I would definitely be open-minded to what kind of form this could take, and what the readout would look like. Because yeah, I think just feasibility is going to be huge for this, because like Avi was saying, there's a lot of confounders about where you live, and what's the regular temperature during the day and stuff like that. So I think feasibility is going to be huge, and yeah.
Okay. Let's see here. Well, if that one is if that one's iffy, then this next one is definitely not gonna.
I think it's great. I think, same with this kind of the drink water idea I had, I think these are important things, right? There's assumption that, like you said, saunas are actually helpful, and and that heat in some way has therapeutic properties, but like, there's not really anyone who's been rigorously studying this. And so I think it's a great idea. I think it's just the devil's in the details.
Okay. Let's see here. So, yeah, okay. I will go through this next presentation, but the expectations are sufficiently dampened. I'm gonna have to share my screen because it is there are slides for this one. Slide, there we go. And we will share screen to this one. Okay. So this is going to be a really I'm not expecting a lot of people to fully understand this, but I'm going to try to explain this as best as I can. We're going to go through the fundamental nature, what I think the fundamental nature of aging is, and it's going to be the schism between germ cells and some somatic cells. I'm going to use a lot of biology terms, so apologies to anybody who doesn't know a lot of biology terms. So we're going to start with germ cells, some background. A germ cell is any cell that has the ability to propagate indefinitely. A lot of times when we say germ cell, what we talk about are the propagation cells for multicellular creatures, but also things like E. coli, funguses, protozoa, protists, stuff like that. Those are all germ cells. And the contention here is that all true life is actually unicellular. What we are as multicellular creatures is actually just this shell, this somatic shell built around the true life, which is a single-celled germ cell. And that with diatoms, it's very easy to see this kind of setup. A diatom will build a shell out of silica. Other cells will build shells out of cellulose or chitin or other things. But then I think at some point cells figured out that they could build a shell out of other cells, and that's where multicellular life rose from. You'll notice that I don't think that there's a single multicellular creature that when it divides, when it multiplies, it stays multicellular. It always passes through a single cell phase, and that's because It's not actually like a true multicellular creature. What it is a single-celled creature that has built this shield, this robot around itself that it uses to make life easier for itself. And then at some point it builds a new robot, and that's how it interacts with the world. Is not directly manipulating things, but rather building a machine that then does all the work for it. And all it does is keep building successive machines. The analogy here is that we are building cars and we're trying to put AI into those cars. And it's the same thing. Like, why do all the work when you can build something that'll do all the work for you? So some more background. What is biological immortality? It's physical continuity over time for cells. Humans will also have to worry about psychological continuity, but that's for another discussion, and it's a little complicated. So for cells and for biology, physical continuity means biological immortality. Okay. Okay. So this animation is basically showing you what life looks like from the germ cell's perspective. So it divides, the egg cell will get fertilized, it'll divide, it builds a shell, it leaves the shell. It goes haploid, it brings sperm in, it goes diploid again, builds a new shell. And it just sort of keeps doing this over and over. And you'll notice that the actual egg never stops being a cell. It never actually dies. That's the orange thing in the middle, the golden cell in the middle. The soma will die. The soma will actually stop existing, but the egg cell never actually stops existing. And it's been doing this for, billions of years. So it's a pretty robust process. So if we think about germline physical continuity, there's a little bit of philosophy here. We think about cell A dividing into cell B and C. If we have a camera, all right, and we focus on cell A, and we take the upper pathway and we follow it to cell B, what we see is cell A ejecting cell C to become cell B. And if we take that camera and we follow cell A down to cell C, we see that cell A is ejecting cell B in order to become cell C. And so cell A and B are physically continuous. There's no denotion, there's no like symbol that is for physical continuity. So I made one up. So it's on the right-hand side there. And then cell A and cell C are physically continuous, but cell B and cell C are not physically continuous with each other. Okay, so for this, when we think about human lineage and human, I guess the progression of our lives, we tend to think of this top diagram, which is if you look at like this, I wish there was like a way to mouse pointer with pen. Okay, you can all see this red outline. Okay, this is a single human's life. And this is a person who has had 2 children, and those 2 children branch off of this in this middle section here. And we think of it as, okay, the children are this other thing, and then they're branching off, and then they have children and they're creating these other things. But the person just keeps living, right? Like, because we think of life from a somatic point of view. We think of life from our soma, and the soma does not branch off. The soma stays put with the original somatic structure, but the germline cells do branch off. And when they branch off, the causality of that branching can be seen of as this structure up here, but it can also be rearranged to this structure down here. Okay, and what life looks like from the germ cell's perspective is it could look like this top portion, but it could also look like this bottom portion where it's systematically constructing new somatic shells and then leaving the old shell. So the way that this whole system kind of works is that, and this is another diagram, it's going through 3 generations on the right hand side, is that there is a initial germ cell it's a loop. So wherever you start in it, is the starting point. You know, it doesn't matter. But so we'll start with a somatic shell around a germ cell in the center. It splits up into a whole bunch of replicants, and this corresponds to pre-birth oocyte production. It peaks about, I don't know, 5 months in or something like that. And then there's an event called oocyte attrition. And that's prior to a woman being born. Is most of the egg cells will die off in a massive event. If we go forward a slide, the best theory that I like for this particular event is this theory I found from a paper called A Role for Retrotransposon Line 1 in Fetal Oocyte Attrition in Mice, where basically the oocytes are attacking each other with retrotransposons. Retrotransposons are just fragments of DNA in the human DNA, where it doesn't seem to have any usefulness. It just seems to be like terrible DNA. But some people think that the egg cells are using these fragments and basically launching them at each other. And the ones that can't deal with these stressors, this genetic stress, will die off. And so anything that has good DNA, repair machinery, everything that's put together correctly will be able to survive this massive die-off. And the ones that are not put together correctly will perish. So it creates this massive replication, and then a bunch of them die off, and that continues to kind of die off over time. And at some point in the process, there's going to be a reproduction. So that's going to be this event over here. So the sperm comes in, there's a controlled mutation, another level of controlled selection. This time it's going to be sexual selection. They're looking for genes that have survived the current environment and are doing well in the current environment. Then the germ cell creates a new shell around itself and leaves the old body. And this keeps happening over and over again. So from the germ cell's perspective, again, the time between controlled mutation events, what it wants to do, the only way it can interact with the world is by building new shells. Okay, so in order for it to interact better with the world, in order for it to have more control over its ability to respond to the environment, it wants this time between controlled mutation events to be as short as possible, which means that, and this is a very old idea, it's actually an idea from the late 1800s, but life is actually trying to compress the lifespan to as short as possible for somatic cells. The germ cells are already immortal, they don't care. But for the somatic cell, the somatic process, it's always trying to compress that time to be as short as possible. And so our long lives are actually an aberration compared to what is actually happening. And for these germ cells, when they look backwards, what they see is an unbroken chain of life. But when they look forwards, strangely enough, they see almost a 0% chance of success, because you're about, this chart's a little bit low. It's supposed to, the numbers that I've heard are about 8 million, but everyone gets different numbers. About 8 million egg cells get created. So that initial cell will split into 8 million egg cells. So the success rate's about 3, right? On average, people have about 3 children because population is growing. So there's 3 successes out of 8 million. That's the chances for any specific division that the egg cell will will be the actual successful egg cell. What I call the protagonist cell, which is the gold line in here, which it has no idea which cell is actually going to make it, but this whole point, the whole system was designed to create as much probability for success as possible. It's kind of an interesting paradox. Anyway, we did that. So we went over this already. Germ cells control, they want to, I said all this stuff. So nature pushes somatic cells to the shortest possible fecundity cycles, and germ cells to the shortest possible fecundity cycles. Fecundity cycle seems to be directly related to lifespan. So now you get this sort of like a lifespan controlling mechanism. So biological immortality is common for germ cells. It's robust for germ cells, and it's natural for germ cells, but it's not common, robust, or natural for us humans. And seems like the obvious thing is that we are just, we're cars basically. And the problem with trying to solve aging is that if you create immortality, you're basically dooming yourself to certain death. It's an interesting system. What happens is that nature is doing this up here. It's creating iterations, right? It's improving constantly. And anti-aging means the stagnation of your body. It means that when everyone else is a roadster, you're going to be the Model T, right? Everything else is constantly going to be evolving, and you're going to be staying the same. Why would a germ cell, which is trying to live forever, ever want to keep the same somatic shell around itself for longer than it has to? That's a dumb idea. It's just really bad. It's just really bad. It's certainly going to fail. Okay. So then the question is, why put effort into regeneration of the somatic system when the shell is going to be obsolete soon anyway? And the some of the good examples here, the Sequoia, which lives for 3,000 plus years, it's super dominant in the forest until these monkeys came along with these blades and just destroyed them, right? Like evolution will annihilate anything that doesn't change. And so the next step is, okay, let's use technology, right? We can get around this by adhering to the fundamental nature of life itself. We get around this by using technology to build shells around ourselves, the same way that the germ cell keeps building shells around itself. We build, cars, planes, boats, personal suits, spaceships, etc., houses, updatable things. Okay. And at this point, now we're the germ cell, right? We're the germ cell that is building shells around itself, and we can adhere to the fundamental logic of the universe while also not dying. We want to occupy the niche of the germ cell. And humans are good at copying, right? So you see a horse running across a field. You say, I want to run across a field really fast. It's very easy. You just jump on the horse. That was a really easy way of copying. Some other stuff a little bit harder. We see animals going underneath the water for long periods of time. We can't go underneath the water for long periods of time, but we build submarines, we build scuba tanks, we build flippers. We can go underwater for long periods of time now. We say, oh, I see birds in the sky. I want to be like those birds. Okay, we don't have wings, but we can build them. We can understand the nature of the birds, how it flies, and then you build these structures and you go into the air. So the question then is, why have we not tried to copy, or why are we not trying to copy germ cells or understand the nature of biological immortality and then copy it? And basically at this point, in my opinion, we're a bunch of medieval peasants standing under a flock of birds, commiserating with each other about how flight is impossible, right? We're like, oh, nothing can fly. It's just impossible. And it's just like, okay. But the examples of flight are all around us. The examples of biological immortality are all around us. Another example is that we're 19th century peasants standing on an oil field. And
what's the biological immortality that's all around us?
Is the germ cells that are constant. They never die, right? They don't age. They don't die. Okay. So we're surrounded by a diverse and almost limitless number of examples of life forms that don't die, but we just don't see it. We're not looking in the right place. And so it's like if you're a 19th century peasant and the only way to get oil was to kill whales, and some guy comes along and they're like, hey, you can just poke a hole in the ground and like 1 billion gallons of oil are going to like shoot up from, and you look at this person, you're just like, what are you talking about? Like, no, you get oil from killing whales. Okay, that's how you get oil. That's the way to get oil. If it was easier, people would have figured it out. All right. So the goal here is, rather than trying to become an expert in aging, to become an expert in germ cell immortality. And the first target here is going to be mitochondria. There's a lot of things, a lot of pieces of aging that can be solved from just looking at germ cells, but intracellular damage, the damage between cells, is probably not going to be one of them. So that might not be a solved problem for germ cells, but mitochondria, genetic instability, other things definitely are solved problems for germ cells. So the idea then is you build a database of single-celled eukaryotes with mitochondria. You compare that to other types of cells. You find patterns, conserved elements, just conserved structures, conserved logical mechanisms in the cells, in the single-celled organisms in the world that are functionally ageless, that they deal with mitochondrial damage. They deal with reactive oxygen species damage. They deal with having mitochondria and also just never dying. How do they regenerate their mitochondria? What mechanisms are being used? There's probably not the same mechanism for every single life form on the planet. There's probably diversity. So now you want to get as much information about the nature of mitochondrial regeneration as possible from these cells. You apply those mechanisms to human pluripotent stem cells. Boom, you get full mitochondrial regeneration, and you don't have to worry about all these other ancillary things happening. This is just an explanation of, your germ cells like the CEO, okay? And the pluripotent stem cells that is like the middle management, and then the fibroblasts are like your workers. Typically, you don't want to give more power to the fibroblasts because that leads to cancer. So if you try to give the fibroblasts massive regeneration, it's generally a bad idea. But for the pluripotent stem cells, that middle management section, if you can figure out how to get them to not age, since they propagate all the fibroblasts, then you're gonna get, I think you can get most of the way there to just solving the whole problem with aging. So this is the end of the presentation. So all we're trying to do is copy mechanisms.
So Aaron, you're in good luck here, because Carlos Bustamante, he has this paper which has this idea. So essentially, it's if you're all not familiar, this kind of Yamanaka cocktail that causes reprogramming. He is introducing this cocktail. It causes reprogramming of fibroblasts or whatever to an embryonic stem cell. He and now other people are basically introducing this cocktail into animals, and the reprogramming to this more germ-like state actually does alleviate a lot of aging-related phenotypes. So this is a really nice Cell paper in 2016. So I think there's been a lot of papers since, and yeah, so I think I can put here in the chat. So this is definitely this actually, I think, is potentially one of the most promising areas of therapeutic areas. And certainly we're trying to do it in my lab and my companies and stuff. So, so I think this is a great idea.
So what other people are doing in terms of Yamanaka factors and trying to turn fibroblasts back into pluripotent stem cells, they're basically working within the framework of they're still working within the framework of simply looking at human cells, only human cells. What I'd like to try to see or to do is to look at just try to search out and treat the single-celled organisms in the world as like a mine, right? You're going to try to mine for information about how the nature of mitochondrial regeneration works, not just in our cells, but in any cell. And then you figure out the mechanisms, and then you can apply those mechanisms. You can pick and choose and try to build something rather than trying to deal with one very specific example. You figure out it'd be the same as like for flight, if you're trying to figure out bat flight and only bat flight, and then you're trying to make humans fly like that, instead of just understanding the nature of flight in general. So you look at every mechanism of flight, you figure out which one's the easiest, right? You figure out, okay, here's how birds fly, here's how insects fly, here's how bats fly, here's how plants kind of fly. And then you see, okay, what can we do, right?
I think, I mean, I get your point. I mean, your point is to principle. I mean, I think you have to reduce it to practice, and so that's what they've done here in this paper. I think it's, but yeah, I mean, I think it's, I mean, obviously it's a great idea. I think it's, but you'd have to chip away kind of organism by organism or whatever to make. So, and I think the idea, I mean, in other organisms, it's kind of, it's kind of already proven, like planarians, right? They have these kind of germ cells, which essentially can, these are the ones that you can chop off their heads, right, or whatever, and they regrow and regenerate. So there are species where, essentially the germ cells are, they're kind of immortal, assuming they don't get smashed by a car or whatever, right? So, yeah.
I mean, technically, E. coli is immortal, right?
Yeah, I mean, yeah. I mean, they're all, I mean, everything's,
right.
Yeah.
The zooplankton are all like single-celled creatures, they have to be functionally immortal. If they weren't, they would just die off naturally. They wouldn't even exist because they couldn't propagate indefinitely. So they have to be able to regenerate indefinitely.
Yeah.
But nobody's looking at that stuff. So anyway, it's just
I would just, I have one thought when you're saying that single cells are immortal. That is true, I think, only to the point where, yeah, one single cell or E. coli replicates, and then you have replicate E. coli that goes on to live forever, and the colony doesn't die, but the individual E. coli unit does die, doesn't it?
Yeah.
I mean,
it's the chronological aging versus the replicative aging. That's best studied in yeast, where they give
exactly
the mother cells gives daughter cells, and yeah. So yeah, I mean, I think the original unit does die. I mean, it has a
when you say original,
so then you can say Homo sapiens is also immortal. Like, I get kids, they get kids, they get kids.
No,
Okay.
so for yeast, okay, when you talk about yeast and budding, when you basically the contention here is that when it buds, that budding can be considered, since it's physically continuous with the parent cell, that's still the parent cell, right? Imagine it squeezing itself into a tiny bud and then just leaving. It's still like that. But for yeast, if you had a yeast system, right, where we had budding, if our pluripotent stem cells did budding instead of symmetrical cleavage division, that right there solves a whole bunch of things, right? If you could have damage segregation mechanisms and you apply it to pluripotent stem cells in the body, and you could segregate damage into one of those, when it divides, it segregates the damage non-symmetrically. That's huge, right? Like, that's a huge idea. You take the yeast cell divisions type and you apply it to a pluripotent stem cell, and now you can get this old part accumulates all the damage, and then you get autophagocytosed, and the new cells are all clean, right? Because that's essentially what you're saying is happening. That's a mechanism that you can apply to the human body, and then all of a sudden things are completely changed for us. So any damage segregation mechanism that people use to explain unicellular aging is still an opportunity for us to use that for ourselves, right?
I don't think what we're saying is in disagreement, though, with this.
Right. It's just that I think it's beyond the purview. If showerheads are beyond the purview of Vita Tau, this is definitely not within your guys' wheelhouse. So
No, I think it's all great. I mean, I think it's just you have to reduce it to practice at some level. I mean, otherwise, I mean, it's great to just debate science, but we have to if you want.
Well, I think that I could build a database. I think I could actually get rid of mitochondria from the conversation with less than $250,000. So just take that as you will, but
What do you mean by that? What do you mean by that?
you build a database, right? It's not difficult. We can get the idea of how mitochondrial regeneration could work by looking at, rather than trying to, go about it the way that other people have gone about it. Instead, you're looking at patterns. So all you need to do is build a database out of cells that have been well studied, and the well studied cells, a lot of them are single-celled, eukaryotic, close to humanized cells, because those are the ones that are pathogenic to us. So a lot of pathogenic species have mitochondria. So now you take a look at the data from all those pathogenic species, you assemble it into a database, you look for trends, you look for conserved elements, and you figure out the nature of mitochondrial regeneration, ideas for that. And then you can, from there, you build, you can just, you can eliminate that mitochondria problem from the entire conversation of aging. Boom, it's done. You figure out the mechanisms, how they regenerate their mitochondria. Then you can apply it to humans.
Right. I mean, you have to be on. A lot of studies start with some kind of bioinformatic thing where you go through and generate a hypothesis, but you have to empirically test it, right? And so,
yeah,
I mean, that's where the rubber meets the road, and that's where all the work needs to be done. So, I mean, and my comment is, it's a great idea, phenomenal idea, but somebody has to do it, and so that's what I think we need to figure out. If you have an idea of who would do these experiments, whether you do them or whether how this would work, I think that's what Beat It Out should be focusing on, like the actual
implementation. Yeah, and Showerhead is not a therapeutic that is actually going to take us very far, but these types of things could. And
I think Showerhead could think.
You're free to
just have somebody has to do it, right? So, I mean, I think these are all both interesting ideas. Just we have to kind of see what they look like in practice. Who's actually going to do them. What the reality.
So yeah, something like that study with Showerheads that can be super useful as a step towards what we really want. It's just not ultimately, it's not going to make too much of a difference.
The heat shock stuff is
something like this. You're definitely encouraged to make a proposal, and why not? I don't think it's outside of our wheelhouse at all.
Okay.
Definitely.
Okay. Let's see here. It's about an hour, so I figured that would take up all the time. So I'm gonna end it here, but I'll do another meeting. I think there's a few more ideas to cover. There's no other low to the ground ideas. So the next one is mostly going to be just a bunch of not necessarily esoteric, but just stuff that would be nice, but it's unlikely that anything would be directly VitaDAO capable. It's just that it's nice to know this kind of stuff in general, to be aware of what the next steps are for whatever pieces of the field that exist out there. So any other final comments from anybody? Otherwise, just gonna, okay.